Hello and welcome to the Breaking Down Biology Q&A session. Today we're joined by Dr. Mustafa Özçam from the University of Maryland. We're going to discuss his research on the association between the gut microbiome and the failure of the peanut autoimmunotherapy. It's great to have you here today, Dr. Özçam.
To be here.
Okay, awesome. So let's start with the inspiration of this research. The peanut autoimmunotherapy has been popular for several years. It's the leading treatment for peanut allergies, and it's been proven effective in many patients, especially young children. However, it surely does not work for everyone, and even when it works, some patients fail to achieve remission, which means that the allergic reactions will recur when the patients stop receiving the treatment. So as we look at the failure of the peanut autoimmunotherapy, what clues first inspired you to connect it with the community of microbes in our gut?
Yeah, I think I should start with explaining or clarifying the failure of the treatment in peanut autoimmunotherapies. You're right that some of the patients did not or do not develop long-lasting remission. Peanut autoimmunotherapy has been shown great success in desensitizing the immune system to peanut proteins, which means that although it did not treat the peanut allergy full, it reduces the allergic reaction threshold or increases the allergic reaction threshold, and many people become more desensitized to peanut proteins after the treatment. To come back to your question, what inspired us to investigate the relationship between gut microbiome composition or the gut microbiome functional capacity and the treatment response in peanut autoimmunotherapies, it really came from the previous literature, where over the last 20 years, many research groups that investigated the relationship between gut microbiome and allergic diseases in asthma, in food allergies, as well as atopic dermatitis, they showed that the gut microbiome is distinct in terms of composition and in terms of functionally between individuals that are allergic or not allergic. And this data was at the level of association over the many years. And then recently, in last 5 years, different research groups again mechanistically showed that gut microbiome is enough to prevent allergic disease development, food allergy development in mice models, where they colonized mice. They did not have any microbiome with a healthy microbiome versus a microbiome from a food allergic children. And then they induced food allergies in those mice. And surprisingly and amazingly, the healthy gut microbiome prevented mice developing food allergies. That really suggested that gut microbiome plays a causal role in the development and resolution of food allergies. At least it suggested that way. And in terms of immunotherapies, again, over the last 10 years, different research groups showed that gut microbiome is associated with cancer immunotherapy, for example. And considering that food allergies are driven by food components like peanut proteins or egg proteins, therefore, we hypothesize that gut microbiome is associated with the failure to treatment in peanut autoimmunotherapy.
That's amazing to see the potential of the gut microbiome and its role in this therapy. So by monitoring and observing and investigating the changes and activity of the gut microbiome throughout the course and after the therapy, one of the most striking findings of your research is that the gut bacteria seem to be able to metabolize the peanut proteins directly. So was this surprising for you to see that the gut bacteria could interact and interfere with the therapy in such a way?
I think for the field in food allergies and immunology, I think it's surprising, but it was less surprising to me because I am trained as a food scientist from my undergrad, and I became a microbiologist and microbiome researcher in early life allergic diseases over the years. And as a food scientist, it wasn't surprising for me that the gut microbiome interacted with those proteins that are giving those peanut allergic children as a treatment. Because again, those treatments, peanut autoimmunotherapy, is peanut proteins, basically, or grounded peanut powder, and they are taken through oral route, and they go through a gastrointestinal tract from stomach to small intestine, and there are millions of gut microbes that are colonized on that surface of the gut, and they are capable of metabolizing many complex proteins. It wasn't known before our research that gut microbes can metabolize peanut proteins. And I think this field is going that direction a little bit in terms of microbe allergen interactions. So no, it wasn't surprising for me. And I think it is surprising for the field.
Yeah, sure. I believe the finding of this research surely provide something that's a little surprising, also very informative for the field. So the final question will be that how do you think this finding will contribute to the future of the peanut autoimmunotherapy or other types of autoimmunotherapy?
Yeah, I can only speak for peanut oil immunotherapy. And then we discussed that in the discussion in that paper, where now we know that gut microbiome is distinct between responders and non-responders. When I say responders, I'm referring to children who develop remission. And then we think that gut microbiome can be utilized to predict who would or would not develop treatment response, because peanut oil immunotherapy is a long treatment. It takes approximately 3 years, at least the treatment that we investigated. And along the way during the treatment, there are some side effects like adverse events, for example, anaphylaxis in some children, and it is stressful to many patients and many families. And by using microbiome, some biomarkers from microbiome, if you can predict who would or would not develop remission, or if you can predict who would fail the treatment, you can think about, can we manipulate microbiome or prepare that patient, specific patient, to make them more likely to develop remission. So prediction is one angle that can be informed by this research. And then another angle is that now we know that gut microbes can metabolize these peanut proteins that are given as a treatment, right? And then that means that each individual might be getting different amount of treatment based on their gut microbiome's ability to utilize those. So first, we need to understand how that is happening. And then second, can we develop a better treatment where we escape from gut microbiome's ability to metabolize those proteins? For example, can we encapsulate them in a structure where that is only reaching the site of action and escaping gut microbiome metabolism, so that each individual, each patient received equal amount of dose at each stage. So that is the second angle we thought could be improved through this research.
Yeah, I would definitely see a promising side and how helpful this research might be in terms of both prediction and also the clinical applications to
Yeah,
give the patients more individualized treatment experience. And as far as I know, Dr. Özçam's lab as well as the Lynch Lab at UCSF both have a research focus on developing next generation microbiome based therapeutics. So it's like this topic is now being actively investigated, and there are surely a lot of people who care about this. And hopefully, we can also see more promising results in future research and clinical trials, and even more approvals for established treatments.
definitely. That would be very exciting.
Yeah. Okay. I think this is a great place to end. Thank Dr. Özçam again for taking the time to speak with us today about his research. To read the article or learn about more information, please check our website, breakingdownbiology.com. See you next time.